repertoires, and a question, how big is it, which means the repertoire. We talked about not recognizing cells, and we talked about actually recognizing pathogens. So this non-recognizing cell thing is important. So a lot of people have thought about this. So let me tell you one more thing about it. Before moving on, there is a point 3. So we will get there. Okay. So only a fraction of the self-peptides get presented in the thymus. So we already sort of mentioned this in one of the questions. Somebody asked about time. Basically, a T cell in the thymus spends a few days, 4 to 5 days. There's no way it can meet absolutely every self-peptide there is. I mean, in a way, you know, no matter how much time it would spend, there would be no guarantee it would meet every self-peptide. So again, that's sort of the textbook picture, that's what they're supposed to do, right? They're supposed to meet every self-peptide, and as long as they haven't recognized any one of them, then they're let out. But clearly that can't be what's going on. So there's a fraction of presented self. Let's call it X. And let's be more specific, this fraction is actually quite small. And this is over the whole time the T cell spends in the thymus. Okay, so if we have R T cells again, as we did before lunch, the number that binds a given self-peptide, so among R T cells, let's call it T cells. Okay, is basically if I have R and I have a probability of this T cell reacting, so let's say recognition probability times, oh sorry, I forgot to say this. So now we're not in the thymus. We're in the, this is real life. Okay, this is the real life of a T cell. We're out actually trying to recognize pathogens, but there's of course some self-peptides around too, right? So the formal word is in the periphery, which basically means blood, tissue, etc. Okay, so this isn't school anymore. This is the real thing. Okay, so it's the probability that it recognizes something times the probability that it hasn't encountered this self-peptide in the thymus. So altogether, this is the probability of successfully recognizing a self-peptide in the periphery, and so this is basically a binomial distribution. Okay, I have R shots, R T cells that are trying to recognize a self-peptide. They have a probability of recognition, and if they've already learned not to recognize one, then that decreases the probability of recognition. I know it's after lunch, after swim, and afternoon, and hot, and sticky. Okay? But basically, you know, It's a yes/no question. A T cell goes up to a peptide, to a self-peptide, and asks, hey, self-peptide, will I recognize you? With probability P, it will. But then it says, have I already seen you? And if the answer is yes, then that decreases that probability. Okay? And it goes up to many ones like that, so that's why it's a binomial distribution. All right. This is number of trials, of T cell trials. And since we're in the limit of having a lot of T cells and this probability being very small, we can approximate, okay, maybe I should ask you, we can approximate a binomial by what? Gaussian. I heard Gaussian somewhere, right? When the question is we can approximate by what, the answer is usually Gaussian. We agree? Okay. And it's a Gaussian with mean of this, right? I'm flipping coins. This is my P. What's the variance of my coin flipping? What's the variance of a Gaussian if I have a probability of success P and failure Q and N trials? NPQ, right? So if you work out NPQ here, given that Q is 1 minus P, you get the same thing. Okay? How did that happen? If you're curious, this is R P1 minus X, 1 minus P1 minus X, but this is so small that it goes away. All right, so these are self. These are our self. All right. Now let's do the foreign ones. Same thing. So again, among our T cells, the number that bind a given foreign peptide will be T, which will be chosen from my binomial distribution with R trials. But now it's just P, right? Because I could not have seen it before. I'm assuming I couldn't have seen it before. I'm looking at the naive repertoire, so there's no problem of training. I've gotten rid of training, and in the same limit, I have a Gaussian. All right, so what does it look like graphically? Graphically, it means that if I plot the number of activated T cells versus probability of activation, I have two Gaussians. Oh, let me look, I've been so good, I've used colors, so let me use the colors. I have myself and I have my pathogen. Right? My idea is that I want to activate more pathogens for the foreign ones. And RP is bigger, so this is RP. This is RP1 minus X. All right. Just to make things clear, pathogen, self. Right. So if I now want to choose a threshold, we talked about this, right? Somebody asked the question about if it, if it binds, is that enough? And I said it's more complicated. So this is how one of the ways it's more complicated. If I'm to choose a threshold here for T star, which will be the threshold at which I'll start to respond, where should I put it? What do I want to do? Yeah. What do I want to respond to these. I don't want to respond to those, right? So here, right? I should put my T star in the middle. So you can frame this as a minimax problem and solve it, or you can wave your hands and cheat and say, well, that's going to be the midpoint between here and here, which is what I will do right now. So the midpoint between here and here requires adding these two and dividing by 2. Hopefully you can trust me that the magic answer is this. Or you can do it in your head. Did I get a minus sign wrong even with that? No. Okay, so let's put it in a box. Doesn't matter, but keeping ourselves awake. Okay, so this is a threshold value for how many T cells should raise their hand and say, yes, I recognize the bad guy, to get a response. Anybody recognize this idea by a name from something else they know about, like bacteria? Quorum sensing, exactly. Okay, so it's an idea for quorum sensing, and okay, and then what is it? So again, nobody really knows, but let's say, I mean, this is actually quite well known because maybe the number isn't well known, but there's a lot of people who sort of think about what peptides will be presented on which MHCs and so on. So the fraction of presented self peptides on any given MHC is about that. And then before that we said that the number of unique T cells is about that. We could instead, so this is the total number of unique, we could instead take 10 to the 6th, which is an estimate of the number of T cells in a lymph node in a given time. So lymph node, or basically it means involved in an infection. Remember, lymph node is where I told you that things get honed when so on. So depending on plugging in these numbers, we're going to get a T star of 10 to the 4 here and a T star of 10 to the 2. You can do, so these numbers are too high. They're not what's seen experimentally, but there have been experiments that have shown quorum sensing in T-cell decision making, not precisely for this problem. It seems T star is really smaller and is around 40. Okay? So let's say in the language we talk about here, 10 to the 2. Right, just for fixing ideas. Well, actually, so this is quite, 10 to the 1, 10 to the 2. So it's not too far maybe from that. But there is quorum sensing. If you're interested in this, the reference is Polonsky in Science, and I don't remember the year, but it's Polonsky, Friedman. And this idea of defining a threshold is in a paper by Butler et al., PNAS 2014. All right. So that's already one complication. Everybody happy with that? Yeah. Yeah. And this paper has the minimax calculation if you're into that kind of thing. Okay? Anyway, they talk to each other. Now, to address another question that was raised, I'm going to sketch something that maybe Thierry will actually calculate in a more general case in detail. Will you? Kinetic proofreading? Maybe. Okay. So hopefully now you know T cells are amazing and they can do everything. So one of the things they do is they also do kinetic proofreading. So I'm not going to talk about this in detail, but we're back to the problem of T cell. Okay, I'm going to, it's T cell, but it's really the T cell receptor. The shorthand for T cell receptor is TCR, and B cell receptor BCR. So I'm going to use that because it's faster to write. So it's TCR, and now we have the peptide MHC, which I talked about in the morning, interactions. Okay. So we have a T cell plus the peptide, and they form a complex. Let's make this one a foreign T cell. It's a foreign peptide. And then we have a self. And they also form a complex. And there's a K on, and a K off, and a K on prime, and a K off prime. This is self. Alright, so far I haven't said anything else, anything that we haven't said until now. And we can define an equilibrium constant for this problem, which I take the concentration of the T cell, the concentration of the peptide, and the complex, and this is related to some free energy of binding. Everybody okay with that? Everybody knows? I mean, I guess I missed day one, but I'm assuming somebody who talked about chemical kinetics wrote down something like this. Yeah. Good. All right. So then C prime to C is P prime to P E. There's no minus sign. Delta F prime, delta F. So in this simple process, we can actually measure this difference of equilibrium binding constants, and if we measure them, it's such that with no kinetic proofreading, they can tell apart a difference of about 1/4. So if I got this right, I think so. Okay, so there's about 3-fold sensitivity. Yeah, I mean, this does assume equilibrium. So far it's equilibrium. So far I haven't done any of that, right? I've just simple process. So can we make this better? Well, as you said, we can try to make it non-equilibrium. So we can start to introduce intermediates. And as we heard this morning, nothing is truly irreversible. which is true. So these back arrows do exist, and they'll actually become important. But if they're smaller, if these forward arrows are stronger, then we get this this cascade. Technically, this is realized through a phosphorylation cascade, which should not surprise anybody in this room who has seen these things before, right? So we add phosphates, and so basically here is phosphorylation, and here is dephosphorylation. And the assumption is that KPK on can't read my handwriting. Right, that the dephosphorylation reactions, which are here, are really much less than Kp or K off. But if we do that, then that means that the Cnth state will now be proportional to P e to the beta delta F to the nth. Right? Basically, I do the same thing I did here, but many times. Right? To get to the first one, I'm assuming that T is abundant, so that so that C scales as P e to the plus beta delta F, and so that gives us, so that means that C1 is that, C2 is then C1, right? So I can, C2 goes as P e to the beta delta F squared, and so on, so that when I get to Cn, I have that. Is that okay? It looks less okay than this morning. Okay. C1 is P e to the belt. Everybody agrees with that? Okay. Then C2 is C1 e to the beta delta F. Well, technically maybe this delta F is slightly different, but I'm not worrying about it. Okay, so then I plug C1. And then Cn, so if you want, it can be a delta F tilde. So if you're really being, then we can have a beta delta F and beta F tilde to dn, KdP to dn minus 1. Okay, if that makes you more comfortable. It really doesn't matter. The point is that I'm able to amplify the signal through this irreversibility. Yeah. It doesn't, I mean, it should be different, but in the hand waving that I'm doing, I initially assumed it was the same, but since I'm not getting my nods, I'm getting nervous. Yeah. I haven't put them in yet. Oh, yeah. Sorry, you're right. Good. Yeah. Yeah. There's a K off. You're right. Right. Okay. So then then I'm wrong. Then it is the same. Yeah. Okay. That's where I got confused. Right. So at each step, it can dissociate with the same delta F. So then it will be the same delta F, because if I assume that Kp and K on are similar, or, well, the same, then it's just 1 delta F. Yeah, thank you. Okay. Anyway, I'm getting an amplification signal proportional to n, right? So whatever I had before, I'm just going to get it to the nth. However, if I now do this ratio again, I'm still going to get the dependence on p prime and p, the difference in p prime and p. So if I do, I can't write here, right? Can I cheat? If I now do n and n here, I'm going to get an n here, but I'm going to stay with my p ratio. Right? I get an n. This is the n I get by, right? So now I can get my 1/3 to the nth, but not more. And more importantly, I still have a dependence on the concentrations. So that means if for some reason the concentration of self is like 10 to the 5th times higher than the non-self, I'm still going to get a response. Right? I have, basically what I'm saying is I have 2 knobs. I have the knob from the cascade, apart from the knob from the measured affinity, but I also have a knob from the concentration difference. And honestly, since viruses multiply, I can easily imagine that they're in such high concentration that they outnumber self. Okay? Yes? Yeah. Well, yeah. Well, no, because I can't go back here. I mean, I am assuming this. But yeah, I'm waving my hands in equilibrium. I'll continue waving my hands in equilibrium. Okay. So what can I do? Let me remove this. And add something else. So I'm going to add S, which is a repressor. So every time I get to step 1 of the cascade, I am going to produce a molecule that will activate this back step of the reaction. Okay? What? No, but it helps you go back. So I put a plus arrow on the back arrow, right? Okay? So why is this a good idea? The fact that the main contribution here to the df is the ratio of these 2 reactions. Well, if I had this strongly, I would have to still take that into account. I would have 3 ways of getting out of the state, and then I would have to work that out. Yeah. Yeah. I mean, again, I could have, the main idea is, I really want to make the point that traditional kinetic proofreading it does assume well, it's not traditional because, well, it is traditional, but it depends on the concentration, right? Now I want to get rid of this dependence on the concentration. And I'm not going to do this in detail. So what I'm saying is, let's introduce a molecule that helps me go back. And why is that useful? Because I'm now basically making it to first base. Anybody can make it to first base. Just, through chance, of course. Hopefully the foreign protein has a higher probability of making that than the self protein. But a self protein will make it here often enough. And so I then want to basically I want to divide this by the probability of making it there, and that probability just depends on concentration. So when I do that, I get rid of the concentration dependence. Right? Okay, now I really have to erase something. So basically now, the same thing there with the Cn will scale as right. I've eliminated the pp prime dependence because C1 because of the C1 step that enhances that. So graphically, okay, pathogen self, right, the pathogen line. So if this is my probability of activation, this is the classical kinetic proofreading picture. Sorry, wrong way. Pathogen self. Okay, so this is classical. Right, high enough concentrations of the self will still result in activation, but with the repressor above some concentration, these start to level off. Did I just draw it badly enough? Yeah, I drew it badly enough, sorry. Right, so they start to level off before that. So this is sorry, I'm getting it all mixed up. Should look at my notes. Okay. Right. And this is also with the repress. So they start to level off, but they level off at different places, and now the self basically cannot reach the concentration needed for activation. Okay. Basically, everything gets renormalized by this first state, which is proportional to P. Well, which is proportional to P delta EF. So we divide everything by P delta EF, and then we get rid of this dependence. Yeah. Nothing. Means I'm tired. Yeah. Where? Without the repressor. Okay. What's the repressor? I actually don't know. Molecularly, what's the repressor? Do you know what the repressor is? No. If you want to know what the repressor is, there's a paper by Gregoire, Alton, Bonnet. I have no idea where. I'm guessing with Ron Germain as last author from probably around 2006, 2007. I can look up the reference. I can look up what the repressor is. Because this scheme has been shown to actually happen in real life. Okay. So that's that. Sorry? That may be a review, but yeah. I'll look up what the repressor is. Yeah. Yeah. No, so to you derive it for each one of them, and then you take the ratio. You no, you take you okay. You take the one with the one with the repressor, you derive it with the repressor, and for the self and the non-self separately, and then the idea is that when you take the ratio, you don't have any dependence on the concentration, but in order to have that, because their concentrations are different, you need to eliminate the dependence on concentration from each one separately. So you only need to worry about one. If you can show for one that it goes out, it goes away. Yes. The threshold for what this is, the length of the cascade for one. What sets the threshold? I don't know off the top of my head. I can. Do you know what sets the threshold for where they level off? No. Yeah. Oh, for the concentration, or for where this? Yeah. That's why I'm waving my hands, because to derive all of it, that would be my 3 lectures. But basically what I want to say is there's a scheme. Most of you have probably heard some version of kinetic proofreading. If you haven't, Thierry will talk about it in more detail. But, you know, you won't. Okay. But anyway, most of you have heard of kinetic proofreading because it's like one of the, you know, important examples in signaling. You can come up with a version with bells and whistles that solves the problem of this concentration dependence. And that is what is actually happening in this system. That's really all I want to say. Yes? Yeah. Then you have no problem if one of them is 0. You're in a limit where basically both of them will make it to here, right? And all this scheme does is it basically says I'm sort of, it's a given more or less that it'll make it here. So all I really want to know is how much this part will be amplified. Yeah. And the concentrations, you can think about that P and P prime, you know, I don't want to say similar, but broadly it's a hard problem. If it's an easy problem, then I could do it with the you know, with this, right? If this is like really 10 orders of, if this is 10 orders or 5 orders of magnitude larger than that, it's a no-brainer, right? The problems start coming when I'm in this regime where the concentration of self is so large that it starts to look, it starts to be in a regime where it would activate the system, right? So you know, for a pathogen, of course, you would require smaller concentrations, but you could still activate it because you have hard things. So yeah, you're somewhat in a regime where the effective action of the two is similar, and then you want to, you know, what kinetic proofreading does is it basically amplifies a small number so that it, you know, makes it, I mean, oh, you know, think about it as thinning the distribution, right? Taking something to the power n. But here I'm saying that's not enough because, okay, that helps, but then this thing, the self can still overcome it by being there in very large concentrations. So is there a way of getting rid of this concentration dependence and really just feeling the difference in the binding? What you want to do is you want to concentrate in the difference in the binding energies and not focus and not be fooled by concentrations. And this is a scheme for doing that.
Biologically, these are all phosphorylations, so this is ATP.
Yeah. Yeah. And so then this thing is called adaptive kinetic proofreading, I think, from what I remember. Okay. Anyway, that was an aside, so let's maybe can I erase this? Yes. Okay. So now we get to point 3 of what we want from our repertoire. We want it to fit in an animal. So we already talked about this, but let's revisit this. And let's talk about real estate problems. Okay. All right, so we know again we have this order of 10 to the 9th unique T cells. As we talked about, T cells are proteins. Everybody agrees? T cells are proteins. And so proteins are coded by genes, yes. So how many genes do we have? 10,000? 20,000. For me, that's 10,000 is good enough. All right. So I'm actually going to be even lenient. I'm going to say 10 to the 5th to help myself out, but 10 to the 4th would make my arguments even better. Okay. And where do your genes go? In nucleus? Anyone? Nucleus? Yes. DNA? I know some of you work on eukaryotic cells, so. Okay, so genes go in the nucleus. And how big is a nucleus? Yeah. Depends on the cell, 5 to 20 microns, which is about 10 to the minus 5 meters if I take 10. And just to give those of you who have never thought about how big your nucleus is, a scale, this is 1/10 of a pixel. Okay? A real life scale. A pixel is 10 to the minus 4 meters. Okay, so T cells are proteins. Proteins are coded by genes. I need to code 10 to the 9th proteins, right? We agree? If I need to make 10 to the 9th T cell receptors, I need to make 10 to the 9th proteins. So I need 10 to the 9th genes. Yes? Unless you reuse genes, somebody said. Yes. So let's convince ourselves we need to reuse genes. So basically 10 to the 9th, 10 to the 5, I need of the order of 10 to the 4 more genes. So if I need that, it means the volume of a nucleus, where was it? Here, right? So this goes as 10 to the minus 5 to the 3rd meter cubed is 10 to the minus 15 meter cubed. All right. So now I stick in my additional, I stick in 10 to the 9th genes to a place where I have room for 10 to the 5. So I somehow need this in additional real estate, which if I now take the cubic root of that, gives me something of the order of 10 to the 3rd meters, which gives me about 1 millimeter. Okay, so basically now I need a nucleus which instead of being 1/10 of a pixel is 1 millimeter. Anybody know how much 1 millimeter is in real life values? Have you ever thought of something that is 1 millimeter big? The answer is a tip of a crayon. Okay? So, if my nucleus now is, let me do it here, is the size of a tip of a crayon, my body, which I will be generous to myself and say I'm 2 meters tall, will be 10 to the minus 3 divided by 10 to the minus 5 is 200 meters. Okay, so 200 meters. Any takers for what 200 meters corresponds to? Distance from us to the beach. That's maybe a bit more. Exactly 4 swimming pools, right? Okay, I have that this is 40 giraffes, one on top of another. Since we're in France, it is also 2/3 of the Eiffel Tower. Okay, it's going up to the second terrace. Okay, this is how big you have to be to make all those genes for your T-cell receptors. Okay, so somebody spoiled my surprise. That's not how we do it. We combine genes. No, you said reuse genes, right? Yeah. So how would you reuse genes? How would you reuse genes? Post-translation. No. Actually, very good, but no. I mean, you have the right idea, right? But what's creepy is it happens at the DNA level. Regulation of genes. I still have to encode them, right? This is why I did this, that I don't have enough room to stuff different things in, right? So I clearly cannot have enough one-to-one templates. So that's why I somehow have to, do what they're suggesting is stick things together. I mean, me as the great designer of the immune system today, right? Obviously, nature did this. All right, so what happens? So the solution is that you combine combinatorics and randomness. It's a process that works for both T cells and B cells. And is called VDJ recombination. So let me get my colors together. So it turns out, so let me maybe here, well, maybe let's do it this way. So So each T cell and B cell receptor is actually made out of, when I've been drawing it like this, sometimes I made the point of drawing it like that. It's made out of 2 arms, which are called 2 chains. This is beta and alpha in T cells, and heavy and light in B cells. These are T cells. These are B cells. Now, each one of them, let's take the beta or heavy, they're made out of 3 fragments, which have been called V, D, and J. J, D, V. Okay? And now the way it's set up is in every cell in your body, you have a number of templates for V, a number of templates for D, and a number of templates for J. They've got numbers, they've got names, kind of like, you know, the enzyme stuff we saw this morning. There's like, you know, but every cell has that. Now, what happens in your B and T cell is the following. First, you randomly pick one V, one D, and one J. So let me just give them numbers just for fun so that J free. Okay. This editing happens at the DNA level, meaning you actually pick one and get rid of the other ones. Yes. Okay. So it's a complicated machinery with enzymes coming together that what they do is actually they'll form a loop. So these are, I mean, they're ordered like this on the genome. It'll form a loop like this, cut here, and you'll be left with whatever's left. The one that's closest to the D that survives gets put together, and so on and so on. Okay? And so as you see, when we do that, unless we pick this one, there'll be some more here. So that means if you really mess up, you have another shot at it. There's some rescue. At some point, they just get they're not activated, and then we go on to mRNA splicing and getting rid of them. Okay, but basically, you know, there's enzymes that come, they form these loops, they yeah. Is that enough detail? Yeah. Okay. So we're here, but the important thing is the DNA has been edited. So now your B cells and your T cells have different DNA than all your other cells. Right? Okay, but we're not done yet. I didn't give you numbers. There's 2D, 12 J's, and I don't remember how many V's. I mean, these are numbers for T cells for, I don't know, around 50 V's, I think. For B cells, there's more D's, right? But less V's. Then, Okay. Anyway, when you get these numbers together at this level, you're left with of the order of 1,000 different combinations. Okay, and this is pure combinatorics. Now, what's our goal? Yeah, 10 to the 9th. Okay, so I still haven't solved my problem. I'm reusing my genes, but I still haven't solved my problem. Any other ideas? What do I do now? There's 4 chains, that's not gonna help me. Yeah, random time. Okay, so this is combinatoric time. Now random time. Now more enzymes come along. How did I draw this? Okay. More enzymes come along and they do what I'm gonna do now. So pay attention. Okay? They delete the nucleotides between the V and the D and the D and the J. And then they come along and they insert random base pairs here and here. So these are called DJ insertions, and these are called VD insertions. And I really insert, so I'm only allowed to insert here, nowhere else, but I can insert whatever I want. I mean, whatever I'm anthropomorphizing this process, right? There's enzymes that come, they form, now the enzymes, they form this huge bridge that does these breather modes like this, and just inserts random nucleotides. Completely random in the sense of non-templated. Okay? No. So this, now as a physicist, you should remind yourself that random does not mean uniform. So there are distributions, but okay. So if we do that, we end up with a final product that does get us to 10 to the 9th different possibilities. And then, as somebody said, I've only done one chain. So in fact, I've done beta or heavy here. Then I can also do alpha or light, which is special. Well, it's the same thing, only it doesn't have the D. So I only have the V and the J and my insertions here. But the process is the same, that it comes from, I mean, they're different genomic templates, but the whole idea is the same. Okay? And we've just made a receptor. Yeah. Yeah. Yeah, so remember how I told you I can make every receptor a mouse can make? I mean, obviously we've diverged, but basically I do have the template, and I put in more insertions than a mouse, so I can make that's part of the reason. But yeah, they are under evolutionary pressure. Again, there is actually a group in Germany that looks at the evolutionary pressure on these genes. I don't have like a punchline to tell you. I mean, some of them are more, they're not like, they're diverse, they're not like completely non-overlapping. So if you want to talk about what the hell it is to analyze them, talk to Maria Francesca. Yes. What do I know about marsupials? No, I mean, I don't have anything to say about marsupials. I can tell you about llamas and cameloids. So, llamas and cameloids have only one chain. They don't have this chain. But they still manage to generate a lot of diversity through basically having more genes or more insertions. But marsupials, I've never seen any data about that. Where does it come from? I mean, again, remember that jawed vertebrates have it. So, so the divergence. Okay, so that divergence. So we directly have a common ancestor with fish for this, right? Now, there's another fun fact, which is that non-jawed vertebrates, so these lampreys, they also have an adaptive, what's called a pseudo-adaptive immune system that logically is exactly the same logic, but the molecular elements, so the genes, are different. So that evolved separately and independently, and that's why it's called the pseudo-adaptive and not adaptive, but logically it's the same thing. So so first of all, such a crazy thing evolved twice at least. Second of all, I mean, that means that the fish ones and our ones are actually phylogenetically tied. Yes. Yeah, so these two is what we were calling R0 before. That's going to code for the receptor. So now your cell only has this is your cell, each B cell and T cell only has this set of DNA. So that's why, first of all, they're unique. All the receptors are the same. And second of all, when they proliferate and make offspring cells, their offspring will have exactly the same receptors they do because they can't make anything else. This happens before transcription. Yes.
Yeah, following up on that. So this is kind of happens at the transcriptional level, like it's those.
This is DNA. This is DNA being edited.
So the different T cells actually have different DNA content.
Yes.
Oh, okay.
So if you want to put this in content, that all our cells in our body have the same DNA, yes, except for your B cells and your T cells.
And there still has to be some constraint that this in the end gives you a reasonable protein, right? Otherwise you could have also just made
Very good.
really random sequences.
So remember how we went from R0 to R, the thymic selection step of testing, and I said, point 1 is whether it'll, bind to anything. If it doesn't make a reasonable protein, that's when it gets eliminated. Okay, now the story, as always, is a little bit more complicated. What happens is first you make a beta chain or a heavy chain with a surrogate alpha chain, and you test this beta chain. If it doesn't fold, you have some probability of rescue, trying again, the thing that I just described with the loops. If that doesn't work, you give up, you don't try the alpha chain. But cells die. Cells die massively in this process. And we can estimate from data, we can estimate the probability of rescue basically from looking at in-frame and out-of-frame data. I can talk a little bit more about that, but yeah, cells die. Juan? Yeah. It occurs when they're generated in the thymus. At the DNA level, when you're making a new T cell, a T cell you've never made before, that's when... How do you create T cells? Okay, this is a process. Okay, there's a process called, can I spell? It occurs right at the beginning, before anything we've ever talked about. Yes. Well, for T cells there is no mutations. For B cells there are mutations. So let's just let's say, okay, let's go back to this. Okay, right now we're talking about where R0 comes from, right? So basically we're talking, before that I said there's this pre-selection repertoire, let's not worry about it. Now we're worrying about it. Where does it come from? What's the diversity of this? It comes from this process. T cells are generated in a process which I may not be able to spell, hemapoiesis. Did I get that right, biologists? Yes. Huh? Hematopoiesis. There you go. So we have precursor immune cells that basically stem cells, hematopoietic stem cells, HSCs, that basically differentiate. Some of them will differentiate into your red blood cells, some of them will differentiate into your white blood cells, so T cells and B cells. So that's when it happens. It happens all throughout your life. This happens when a T cell is produced. Okay, then it goes to the thymus. It is in the thymus. In the thymus, it goes through this exam process. Do you bind to anything at all? Do you bind too strongly to self-proteins? That's how we get the naive repertoire. Okay, but we get it from this, which is made of this. After that, and already in the thymus sometimes, can be a few differentiation steps. However, when you're in the periphery and you're a T cell that just encountered the pathogen, you're going to proliferate. You're going to make many copies of yourself to again make this army to basically kill the enemy faster. Okay, and then it's going to go down. So if you look at infection dynamics and T cell or B cell counts, it does something like that. Okay, time count. Now, for T cells, that's basically the whole story. Then they get put into this memory repertoire so that this response happens faster. For B cells, when they proliferate, they also somatically mutate the receptor. What does that mean? I need the new color. So not to confuse it, so that you don't think it happens there. This is now, let me redraw my receptor. Okay, so V, D, sorry, VDJ, okay. Let me just draw one chain. Now we're again in the blood. The naive T cell versus pathogen. Sorry, naive B cell, I was saying, versus pathogen. Okay, in periphery. Okay, so we have this receptor. It's on a B cell that's just recognized the pathogen. It means it's pretty good, right? What does that mean? That the KD, the binding constant to this bit of pathogen, is pretty good. The cell starts to proliferate, so we have a cell with this thing. It starts to make offspring. Again, if it was a T cell, all these receptors would be the same. For a B cell, what happens is that random nucleotides will mutate. Okay? That means if I had, I don't know, a T here, maybe it'll go to an A. Right? Anything can happen again. Again, there's some rules, there's some distributions which we've quantified and so on, but in principle they just mutate in different places. So now I had something that works. I start to mutate it, it may not work again, right? I'm breaking stuff, but I'm also maybe making it better. That's the goal. So it goes through many rounds of this, then being tested again, whether it binds, not in the thymus, but now in the lymph node, whether it binds better. And the ones that bind better go back and get mutated again and over again for 2 weeks. And during these 2 weeks, you're constantly spitting out candidates that are better than the original one and using them. And at the end of it, you keep some sample of what you've produced, but not necessarily the best ones. Yes? You have this bit of, so this thing initially recognized some bit of protein. You have this bit of protein in the lymph node and you're testing against it all the time. This is like a whole story. There's a part where you mutate and a part where you test, and you go over and over and over again. And there's T cells involved, because if suddenly you start recognizing yourself too strongly with them, that's not great news either. But anyway, just to, yes. No. I mean, non-coding part. I mean, this? No, this you really, I mean, like in your liver cell, you have all of this. It's untouched, it's downregulated. In your T cell and B cell, this gets edited and you just end up, okay. Actually, your question again is deeper, and maybe I should explain it. So, remember we have 2 chromosomes. Everybody remembers that? Yes? 1 from each parent. So, of course, you have some version of this on each chromosome. Now, okay, probably you're blown away because this completely makes absolutely no sense, right? I mean, you're putting stuff together, stuff that maybe was okay because maybe the templates were okay, but even if it was together, then you're then deleting and inserting stuff, right? What's the probability that by doing that you'll make something that makes sense? Very low. Exactly. We can actually quantify that probability. But it's very low. So you do that and maybe it doesn't work. Maybe in this step of, do I bind everything, I fail miserably. And that happens. And if you've finally given up on like chewing on this and editing this, well, you go to the chromosome from the other parent, right? Simple. So you have parent number 1, parent number 2, and only if both parents fail you do you chuck the cell away. Okay? So why is this important? Because then if we go in and our experimental collaborators can sequence this thing, can go in and read the nucleotide content of this, right? And if you go in and you read the DNA, well, it makes sense that you're going to find the ones that are actually functional and make proteins, like have no stop codon, right? When you read out genes, there's a special combination of 3 letters that tells the polymerase to stop, right? They won't have that. They'll look like sensible proteins. However, some of them will have stop codons, or will be out of frame. Like basically, by the time you get to the J, you have something that is not making a protein. You have a stop codon here, and it just falls off. Those are things that you're seeing in the data, in the DNA, that's there in the DNA, but could not have ever been expressed and make a protein. So those out of frame ones, as we call them, it means out of DNA, out of reading frame. Basically, those we know, they came from the situation where you first tried with one chromosome, that didn't work, and then you had to move on to the second one. So the cell is there because the second one was functional. Well, in frame, and more specifically functional. So that's why you see it, but this was never expressed. So why is that useful? Well, because if we analyze these, we know something about the statistics of this process. Because the in-frame ones were selected upon. They went through this thymic selection. They had to deal with F. These ones stopped at the R0 level. Okay? And in fact, so remember when you read out DNA, you read it out in bunches of 3 letters, right? Codons, code for amino acids, 3 letters at a time. I didn't make it clear, but when you do this deleting and inserting, you can delete a different number than you insert. So that's why when you get here, you can end up with something that's no longer, You can play this game with words. Take 2 words, delete the end and the beginning, and see whether you can make another word that makes sense out of it. Yeah. It's a very good question. They're only put in here. Now, other funky things happen. What? Is there a cost to only putting them in here and not elsewhere? Okay, this is specific. The enzyme has to attach somewhere. It's basically evolved in a way that it can happen only there. Now you're asking, why has it evolved that way? I don't know. Other funky stuff can happen. So what can happen is you can get two Ds, which again is not supposed to happen, and you can get reverse Ds. Okay, so that you can actually understand why that could happen, because remember we're making loops, and maybe somehow, sometimes, I'm out of, where's my V color? And you know, I've just done the V before and it should sort of connect with that, but what if the V somehow, what if the V does something weird, like, I don't know. I mean, you can imagine the DNA turning in such a way that the D ends up backwards. It does happen, but it doesn't happen that you get these insertions elsewhere. I don't know why. Yeah.
The variability is more inside. Is there a kind of molecular
Yes. Yes, thank you. Okay, this may help me actually. Okay. In my brilliant drawing abilities, let me attempt another rendition of the receptor. This will probably not work, but okay. So, I mean, the receptor looks like something like that. And as we, we draw the peptide here, what actually happens is that this end, and this also can answer another question that maybe some of you have, is if I'm adding stuff here, don't I have, like, even if it's in frame, don't I have a structural problem? A protein needs to fold, right? So what happens is that this variable region is actually at the end here, and is the part that interacts directly with the peptide. In immunology, it's called the CDR3 region. I'll write it for... And it forms a loop. Structurally, it forms a loop. And it is the most important region for recognizing the peptide. I mean, there's other regions here too that matter, and of course, as always in proteins, a non-local mutation can influence, this action at the distance, allostery, and so on. But this thing is really the thing. It's like, the hands that are touching the fact. So that's the reason why selection pressure may be strongest on this region, and that's why you want to generate the most variability here. And you've just killed one of my punchlines about where is the greatest variability and diversity and so on, but we'll still go through it. So, okay. It's time to break again, I think, right? 5 minutes. Questions? I'm going to recommend 2 books to you. I can do this online. One is a book by Carl Zimmer, and it's called She Has Her Mother's Laugh. And this is like Camilleri science version, okay? It's really a smooth read, super fun. And it talks a lot about mosaicism, right? You know what that is? Like when some people have half of an eye a different color than the other, they also have different DNA there, and how that comes about and all, it's just a very, very fun book that talks about a lot of genetic stuff. It's, a beach read. Another one that may actually get in your question is a recent book called Eve, by, I think, Kat Buchanan. I may be spelling this wrong. And this is about the evolution of the female body. and how basically the fact that we walk up straight is a very, very bad evolutionary idea if we want to propagate our species, and how that imposes a lot of constraints. So I would guess that being 200 meters tall is also not like the greatest idea in terms of, you know, being stable and stuff like that. So what I want to do next is basically try to tell you a little bit how, you know, how we can quantify this, how we can actually learn from data, how we can learn from these out of frame things. And then, you know, a little bit about what happens next. Now, part of the things I wanted to maybe do, because it's a school, and I feel pressure to give you some actual like formulas and stuff, is the way we do it is we do an expectation maximization algorithm. So that's a part that's like, that's really no fun, right? I'm not gonna, you know, there's not gonna be mice, there's not gonna be elephants, there's not gonna be anything, there's just gonna be derivations. So how do you feel about that? And first question, does maybe everybody knows what expectation maximization is? Does who knows? Okay. Who wants to know? All right, so I'm gonna do it. Yes? You're gonna suffer?
Maybe you could also provide additional material that we can put on the shared drive if you feel that it's technical and might be bad for the. It's up to you, but there's the possibility of having it.
So okay, so for technical material, I mean, I think we have again, I've mentioned this today. Grégoire, Thierry, and me wrote a review which is called what? Quantitative immunology for physicists? Yeah. It's 2020 physical reports, I think. There's an archive version that is up to date because you have to pay for this, but the archive version is exactly the same. Basically what I'm talking about until now in some form or another is all there. And what that has is also it has technical appendices. So like all derivations, there's like a primer on population genetics, a primer on optimization, a primer on stuff like that. So why don't we sleep on it and decide what I do? Because yeah, I mean, it's not very long, but it is technical. And the alternative is, I mean, I'm gonna finish with this story, and maybe there won't be a question. Then I want to tell you about selection, and then I want to tell you about diversity, and then what I could. Well, I think there I'll stop because the other thing is also technical. I'll give you a reference. And then I could tell you a little bit about optimal immune systems if there's time. Does that sound good? Yes? Okay. If there's any requests, give them to me in the next half hour.